Hello, everyone, and welcome to today's webinar titled Confidence in Your Calibrators, Introducing the MassTrak Endocrine Steroid Calibrator and Quality Control Sets. This webinar is part of the Clinical Diagnostics and Research event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Waters Corporation. For more information about the sponsor, please go to waters.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find this live chat located on the right-hand side of your screen. You can also participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located to the right of the abstract. This survey will be available during both the live sessions and on the on-demand period. If you have any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dominic Foley, Principal and Scientist and Clinical Applications at Waters Corporation. Dominic, welcome. You may now begin your presentation.
Thank you. My name is Dominic Foley, and I'm a Principal Scientist at Waters. Welcome to this presentation on Confidence in Your Calibrators, introducing the MassTrak Endocrine Steroid Calibrator and QC set. This presentation will introduce you to a new IVD steroid hormone calibrator and QC set to help laboratories in their routine LC-MS analysis of steroid hormones. Before I take you through the slides, I would like to highlight some key takeaways from this presentation. The MassTrak Endocrine Steroid Calibrator and QC sets provide metrological traceability that aid laboratories in their compliance to ISO 15189. They also provide improvements in laboratory efficiency through the provision of lyophilized materials. In addition, there is greater confidence in the accuracy of steroid hormone results, and this provides a pathway to laboratory method harmonization with excellent lot-to-lot reproducibility. This slide shows the outline of what I plan to discuss today. Prior to discussion of the Calibrator and QC sets, I thought it might be useful to provide some brief background on steroid hormone metabolism and analytical challenges associated with steroid hormone testing. This will be followed by an introduction to the MassTrak Endocrine Steroid Calibrator and QC sets with some of the associated product characteristics such as metrological traceability. I will then finally demonstrate the use of the calibrator and QCs with an in-house LC-MS method to provide an example of method performance using these products. Before I provide an introduction to the calibrator and QC sets, I thought it would be useful to provide a bit of background into the steroid metabolism and challenges associated with steroid hormone testing. So this slide here demonstrates the key steroid hormones and associated enzymes that play a role in steroid metabolism. Dysfunction in steroid enzymes results in significant increases or reduction of this respective steroid precursors and products. One example is the reduction of 21-hydroxylase activity, resulting in significant increases in its precursor, 17-hydroxyprogesterone or 17-OHP, and steroids from alternative pathways such as 21-deoxycortisol. There is then an associated decrease or significant reduction in cortisol concentrations. Analysis of the associated steroid concentrations can provide an insight into the function of these pathways. In order to meet the requirements of a method to provide insights into steroid metabolic pathways, there are a number of analytical challenges that need to be met. Analytical sensitivity at low physiological concentrations from some steroids, such as testosterone, is required in order to make the method applicable across pediatric and female reference ranges. Selectivity is also problematic too, as many steroids are structural isomers. This tends to cause significant problems with some analytical platforms, such as immunoassay, where cross-reactivity of antibodies can result in problems with precision and accuracy. Although not exclusive to steroid hormone analysis, the preparation of in-house calibrators and QCs can also be a challenge in terms of time and resource. The process in generating in-house calibrators that provide acceptable data for external quality assurance samples can take time. This can be particularly difficult with large numbers of steroids that can often be found in a single panel for analysis. In addition, demonstration of metrological traceability may be required to aid compliance with regulatory guidelines. A wide dynamic range for some steroids, such as 17-OHP and testosterone, is required due to the impact of working across different reference ranges, which include those associated with steroid metabolic dysfunction. In some instances, sample volumes may also be limited, but the analytical sensitivity requirements of a method will still need to be met. So many of these challenges can be overcome using LC-MS, which is why recommendations have been made to use LC-MS for steroid hormone analysis. So in regards to these recommendations, there are some guidelines that I wanted to highlight, particularly two pieces of information found in this slide. One of which is from CLSI C62-A, which highlights the superiority of LC-MS methods over HPLC with optical detection and immunoassay, and this is in regards to selectivity due to the orthogonality of LC separation and mass detection by mass spectrometry. Secondly, in regards to steroid hormone analysis, the Endocrine Society also highlighted the limitations of immunoassays due to the selectivity issues and have indicated that measurement of steroid hormones by mass spectrometry is preferred. So going back to the metabolic pathway, this slide highlights the challenges associated with developing an accurate and precise steroid method. All the steroids shown in this slide are based on the same A to D ring structure, with small changes in the hydroxyl group positions, side chains, and double bonds. These structural similarities are what can cause issues with antibody recognition on immunoassay platforms. Mass spectrometry can provide greater selectivity through mass detection. So here is the steroid pathway with the associated masses of the steroid hormones. We are able to differentiate some of the steroids through precursor mass alone, but multiple reaction monitoring in tandem quadrupole mass spectrometry is also required to provide greater selectivity of the isomeric species. However, even then there are issues with some interferences from the isomers, with mass spectrometer struggling in terms of differentiating between steroid isomers based on the precursor and product for the steroids. So those that share the same precursor can often provide similar product fragments, and these are shown in the same colour in the slide here, just to give you an indication of the complexity involved. And in some cases of steroid hormone excess, you may also find that isotopic forms of the steroids, so the 13C3 forms, may also interfere with steroids that have a mass 2 Daltons larger. So there are potential interferences in the steroid pathway, and these are also grouped by colour. One example is the excess of androstenedione concentrations, and these can appear in the testosterone MRM transition as a result of this isotopic contribution. So in order to work around this issue of these interferences, the orthogonality of LC separation provides resolution of these isomeric steroids. This slide demonstrates an example of that. So by utilizing the correct column chemistry, critical separations of steroid isomers can be performed, which enhances the method precision and accuracy. The examples above are typical of the separation required for LC-MS analysis of steroid hormones, with separations of 11 and 21-deoxycortisol and corticosterone, separation of 17-hydroxyprogesterone and 11-deoxycorticosterone, and even separation of testosterone and epitestosterone. So LC-MS methods in clinical laboratories are often based on laboratory developed tests or LDTs, and these are generally validated to local regulatory guidelines. So these guidelines are constantly evolving, and there is an increasing demand for all aspects of clinical methods to comply with these changing regulations. This includes the Calibrator and QC materials used to generate and independently check the accuracy of the calibration within the method. So there is a growing need for metrological traceable calibration materials to replace in-house prepared materials in order to aid compliance with these guidelines, such as ISO 15189. So what is metrological traceability? So in the context of LC-MS methods, it is a tool to ensure accurate results. It is a process that relates measurement values to a reference standard and a property of a test result, which is maintained through monitoring and correction over time. So in the pyramid shown to the right in the slide, this shows the level of traceability typically applied to analytical methods, where a sample measurement can be traceable back through working and primary standards to a higher order reference standard, such as NIST. So far, I have highlighted some of the challenges associated with steroid hormone testing. Now I would like to introduce you to the MassTrak Endocrine Steroid Calibrator and quality control sets, which can be used to help meet some of these challenges. So this is the MassTrak Endocrine Calibrator and quality control sets, and these contain 12 steroid hormones in lyophilized serum. These include progesterone, 11-deoxycorticosterone, corticosterone, 17-OHP, 21-deoxycortisol, 11-deoxycortisol, cortisol, DHEA, androstenedione, testosterone, DHT, and DHEAS. There are 7 calibrator valves in total, which also includes a calibrator 0, and 3 QC valves at low, medium, and high concentrations that span the calibration range. The Calibrator and QC concentrations span the expected reference ranges for the steroid hormones, in addition to supraphysiological concentrations that can be observed in cases of reduced enzymatic activity in the steroid pathway that I've described in the previous slide. So the Waters MassTrak solution features metrological traceability, lot-to-lot consistency, in addition to controlled and documented accuracy with a CE IVD mark. This delivers benefits by easing the path to obtain ISO 15189 certification. It also enables longitudinal results comparisons and reduces the lab resource burn through being ready to use and implement. So the MassTrak Endocrine Steroid Calibrator and QC sets have been designed and manufactured to assist laboratories in their compliance with ISO 15189. So a key benefit to this process is the improvements in laboratory efficiency, and this is gained from using the ready-to-use, following reconstitution with water, metrological traceable Calibrator and QC materials. So the table to the right highlights the end user steps that are negated by using manufactured provided Calibrator and QCs. And this results in significant time and resource savings that can be associated with the generation of in-house calibration materials. So just going through this list, just to highlight some of those steps, the lab no longer needs to source their own CRM and matrix for the steroid hormones. Gravimetric preparation of standards obviously is no longer required, and calibrators are also prepared, and these are no longer required to be done because the MassTrak Endocrine Steroid Calibrator set is already provided. And in addition, there are stability, accuracy, and lot-to-lot reproducibility of the product is covered, so this is taken care of. And finally, a measurement of uncertainty can also be provided too. So the metrological traceability associated with the MassTrak Endocrine Steroid Hormone Calibrator and QC sets is split across two processes. So what we have in this slide is 8 of the steroid hormones, and these are primary in-house standards prepared at our manufacturing site, and these are traceable to the CRM, so the NIST material, with the values based on gravimetric assignments for androstenedione, 11-deoxycortisol, 21-deoxycortisol, corticosterone, 11-deoxycorticosterone, DHEA sulfate, DHEA, and DHT. And then the separate process covers 4 of those steroid hormones, and these are secondary in-house standards for cortisol, testosterone, 17-OHP, and progesterone. And these are traceable to the University of Ghent and RFB reference measurement procedures. So those values are based on a secondary reference measurement procedure assignment. So now we're moving on to more of the data associated with the product. So data on the accuracy of the calibrator set was generated through the evaluation of in-house QC and external quality assessment samples, so EQA samples. And so we have 3 lots of manufacturing material for the calibrators, and these are evaluated for accuracy and mean deviations from the assigned values at low and high concentrations. And this was following analysis with an in-house validated LC-MS method, and the results of which are shown in the figure. So this approach not only demonstrates the accuracy of the calibrators, because we're seeing deviations within plus or minus 9.3%, and by doing that we're demonstrating the lot-to-lot reproducibility of the manufacturing process. So this is important if laboratories seek to maintain their harmonization standards over time using different manufacturing lots of the MassTrak product. And as part of this overview, I also wanted to just reinforce this slide, which shows the likely suitability of using the MassTrak Endocrine Steroid Calibrator and QC sets in context of the Waters IVD tandem quad MS systems. So they are the Xevo TQD, TQS Micro, and TQXS. So depending on the sample volumes and sample preparation, it is likely that the DHEAS, cortisol, androstenedione, and testosterone and 17-OHP, as well as 11-deoxycortisol, will work across all three systems, whereas to enable quantification of the other steroid hormones, so 21-deoxycortisol, corticosterone, DHEA, DHT, and 11-deoxycorticosterone, as well as progesterone, a higher end system will be required, or is likely to be required. So just to emphasize, as part of the manufacturing process, the calibrators were assigned using the Acuity UPLC I-class with Xevo TQS macro system. So we would typically advise a system of equivalent sensitivity will be required to accurately determine the low concentrations across all 12 steroid hormones. To give you an idea of the performance of the MassTrak Endocrine Steroid Calibrator and QC sets in a lab setting, we developed an in-house LC-MS method to determine the 12 steroid hormones in a single method. So this method is based on an automatable protein precipitation and solid phase extraction format. Internal standard was added to the samples, so 125 microliters of the lyophilized serum, and this was precipitated with methanol and diluted with water prior to centrifugation. An Oasis MAX microelution plate was conditioned and equilibrated prior to loading of the supernatant to the SPE plate. 10 microliters of the supernatant was added directly to the collection plate. This ensures that some of the recovery of DHEAS sulfate is recovered through the extraction, which can be lost through the SPE. And this will maintain the selectivity of the extraction and therefore analytical sensitivity of other steroids in the final extract. Low and high pH washes were applied, and the samples were eluted with 60% acetonitrile, already containing this 10 microliters of sample. 35 microliters of the 50 millimolar ammonium bicarbonate is added prior to injection, and the presence of this ammonium bicarbonate improves the peak shape for DHEA sulfate. So using the Acuity UPLC I-class FDM, separation was performed with mobile phases of ammonium fluoride in water and methanol, and a Cortex C8 column, which is 100 millimeters in length. Steps in the gradient solution were applied to tease apart the isomeric steroid species. Detection was performed using the Xevo TQS macro system, and the MRM parameters shown here should be transferable to the Xevo TQ-XS. The retention time windows are also shown in the table, and any LC flow is diverted to waste outside these overall retention time windows. So moving on to some of the results. This slide illustrates the chromatographic separation of the 12 steroid hormones. So separation of some of the key isomers, which we previously described, is achieved. So we have separation of 21-deoxycortisol, corticosterone, and 11-deoxycortisol, in addition to the 11-deoxycorticosterone and 17-OHP separation. So these chromatograms also show an example of the sensitivity of the low calibrator from the extraction, with signal to noise greater than 10:1 for all the analytes. So moving on to calibration and analytical sensitivity, the mean correlation coefficient of the calibrators and mean signal to noise of the Cal1 calibrator from 5 analytical runs are shown in the table here. So we have some mean R-squared values which are greater than 0.994 across the respective calibration ranges for the 12 steroids. And the signal to noise of the Cal1 was greater than 10:1 across the 5 runs. So this does demonstrate that the linearity and analytical sensitivity of the method can be achieved using the MassTrak Endocrine Steroid Calibrator set and an in-house methodology. So now we can move on to some results around method precision. So total precision and repeatability were determined by extracting and quantifying 5 replicates of 3-level QC material per day over the 5 separate days, so 25 replicates in total. So we have total precision and repeatability of the 12 steroid hormones across the 3 concentrations of less than or equal to 7.7% CV, thus demonstrating excellent method precision of the LC-MS method. And now moving on to some aspects of method accuracy and EQA bias. So we have the accuracy of the QCs, which was evaluated in comparison to the calibrators over the 5 analytical runs. So the mean accuracy for the QCs, or the quality controls, across the 12 steroid hormones at the 3 concentrations ranged from 91 to 112%. Accuracy was also assessed for DHEA sulfate, cortisol, androstenedione, testosterone, 17-OHP and progesterone through the analysis of EQA samples from UK NEQAS. So the data obtained was compared to the mass spectrometry method mean for the samples, all 4 except the ALTM for progesterone, as LC-MS method values were unavailable for this analyte. And also we performed an Altman-Bland agreement on the data set. And this agreement for DHEAS, cortisol, androstenedione, testosterone, 17-OHP and progesterone provided a mean method bias that was within plus or minus 7.8%. And this does demonstrate excellent agreement with the EQA method values or method concentrations for the steroid hormones. So in summary, the MassTrak Endocrine Steroid Calibrator and QC set for in vitro diagnostics contains metrological traceable calibrators which aid laboratories in their compliance to ISO 15189. Significant savings in time and resource can be made by using these pre-made metrological traceable calibrator and QC materials. In addition, the MassTrak Endocrine Steroid Calibrators are reproducible and accurate across manufacturing lots and cover the relevant ranges for each of the steroid hormones. And using an in-house LC-MS method, we can demonstrate excellent reproducibility and accuracy of the MassTrak Endocrine Steroid Quality Control Set. And in addition, we demonstrate excellent bias and accuracy for the EQA samples. So there are some acknowledgements I would just like to make, but generally across the Waters sites around the world, there are different colleagues who have made huge contributions in bringing this product to market. So I'd just like to thank those for contributing to getting this product to where we are today and basically delivering benefits to our customers using this product. And so that is the end of the presentation today. So if you wish to find out more information, you can go to the product page on the Waters website. And this can be found in the clinical consumables and automation section on the clinical diagnostics landing page. So if you'd like to ask any questions, then feel free to do so now in the Q&A. Or alternatively, you may want to contact your local chemistry account manager who can field those questions and provide you with any further information. So I'd just like to thank you for your time, and I'd just like to pass back to the moderator now to help with the Q&A session. So thank you.
Thank Thank you, Dominic, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. We already have some great questions coming in. Dominic, are there plans to include internal standards with the calibrators and quality control sets?
Thanks for the question, Susie. During the development of these calibrators and controls, there were discussions about including internal standards, but in the end we decided there might not be any value to the customer for those internal standards, maybe from a cost point of view. And we know that it is easily it's a case of being able to get hold of these internal standards relatively easily from other commercial means to then generate an internal standard working solution for these steroid hormones. So we decided to not include internal standards as part of the product in this instance.
Thank you so much. And our second question coming in, the method to demonstrate performance of the calibrator and quality control set used solid phase extraction. Are there possible alternatives that could be used for sample preparation of these steroid hormones?
Yeah, so we commonly use solid phase extraction techniques for our steroid hormone analysis. We know that they do provide fairly clean samples and provide a concentration effect to improve analytical sensitivity. There are possible alternatives using, say, liquid-liquid extraction, and we have demonstrated a liquid-liquid extraction sample preparation technique for steroid hormones found in this calibrator and quality control set. So it is possible to do it that way.
Wonderful, thank you so much. And we have a final question. Are there plans to include any other steroid hormones such as estradiol?
There have been some conversations around this. So we know that estradiol in particular is something that is commonly analysed in clinical labs. But we thought in this instance it might be worth having a dedicated calibrator and quality control set for this at some point in the future, but not in this kit at this moment in time, as we didn't see added much value to the creation of maybe steroid hormone panels using the kit in its current form. It is something we are closely evaluating. And the same is true for aldosterone as well, which is another steroid hormone of interest. So we are keeping our ear to the floor and just making sure that we are taking that feedback from customers and seeing if there is any value in including other steroid hormones in our products in the future.
Dominic, thank you for your presentation today and for important research. Do you have any closing remarks you'd like to provide before we go?
So I'd just like to thank everyone for attending the session today. We really value any feedback. So if you like to maybe contact your local chemistry accounts manager regarding this product, if you are interested, then that would be really appreciated. And if you have any interest in finding out more information on this product, then feel free to go to the Waters.com website where you'll be able to find that information.
Well, thank you again for your time today. And before we go, I also want to thank our audience for joining us today and for their interesting questions. And I also like to thank our sponsor, Waters Corporation, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until November 2023. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, Take care, everyone. Stay safe. Bye-bye.